We now live in a world where you can routinely sequence entire genomes from samples collected in the wild. I was kind of a hotshot as a grad student because I had multiple genes included in my dissertation. And my grad students look at this today and they laugh and they said, it would take you a long weekend to collect these data, right? And they just don't understand how much more laborious it was back in the day to do that.
From the heart of The Ohio State University on the Oval, this is Voices of Excellence from the College of Arts and Sciences, with your host, David Staley. Voices focuses on the innovative work of arts and sciences faculty and staff. With departments as wide-ranging as art, astronomy, chemistry and biochemistry, physics, emergent materials, and mathematics and languages, among many others, the college always has something exciting happening. Join us to find out what's new now.
Joining me today in the ASE Marketing and Communications studio is Bryan Carstens, professor in the Department of Evolution, Ecology, and Organismal Biology at The Ohio State University College of the Arts and Sciences. His areas of expertise include phylogeography, molecular ecology, and systematics. In 2025, he earned the Ohio State University Distinguished Scholar Award. Dr. Carstens, congratulations and welcome to Voices.
Thank you.
Well, and I want to start first with phylogeography. I've never encountered this term before. What is phylogeography?
Well, so phylogeography is a relatively new discipline that started right around the late, 1980s, 1990s. And it came about due to a big technological advance that happened with DNA sequencing technology. And so this advance basically for the first time allowed any researcher that had reasonable amounts of resources to go out and collect genetic data from wild populations. And what some of the first people did with this technology is they tried to understand how geographic ranges of species influence the pattern of genetic diversity that they found within those species. And, this was a sort of very popular way for organismal biologists who might be particularly interested in a given taxonomic group to apply genetic data to the work that they were doing. And so you had people who were interested in frogs or birds or plants or bats or whatever, collect data and interpret how the geographic data and the diversity within it were distributed on the landscape. And so some of the early insights that happened were the realization that in places like Europe and North America, most of the genetic diversity was in the southern portion of these continents rather than anywhere else. And the reason for this was that during the Pleistocene glacial episodes, all of the northern portions of the continents were covered by a kilometer of ice, and so nothing was living there, right? So this isn't surprising, but the genetic data and the patterns geographically really reflected this recent evolutionary history.
So what do you work on specifically, or what does your lab work on?
So my lab specifically is, we like to think of ourselves as using genetic data and other sources of data to understand the recent evolutionary history of species. And so systematics is a discipline that broadly is understanding how different living things are related to one another. The only figure that Darwin included in his book on the origin of species was in fact a drawing of sort of a conceptual tree of life. And in the 150 years since Darwin wrote on the origin of species, biologists have been gradually fleshing out what that looks like. Today, our main source of data about how organisms are related to one another is genetic data. And that's just because there are billions of potential data points out there. And like all good scientists, we like as much data as we can get.
Do you work with specific species or?
Well, so I am a bat biologist by training.
Bats, okay.
But I'm really a geneticist. And what that means is that in my lab, I have some students and postdocs who work on bats. I have others who work on other kinds of organisms. I say as long as it has DNA, we're good.
When you say related to each other, relations to each other, you mean like genetically related?
Yeah, genetically. Yeah. Well, and all relationships in biology are a function of that in some way or another. Yeah. Mm.
So walk us through collecting genetic materials in the wild. What's that process involved?
Well, so there's several ways that you can do that. I am affiliated with the Museum of Biological Diversity on campus at OSU. And in that museum, we have millions of specimens that in large part have come into the museum because a researcher has gone out and done a targeted field study. They've either gone to a particular part of the world and said, I'm going to collect all of the mammals or birds or something that live in this part of the world. Or they have said, I'm really interested in this particular species, so I'm going to go to different field sites throughout the range of that species and collect specimens from them. And these specimens are to biology what books are to a library, essentially, right? They're the primary data point. And so in my lab over the summer, I have a team of undergraduate students who have been preparing samples. We have a freezer full of dead bats that were collected from a wind farm, and some of the students are processing those bats and extracting muscle tissue and organs like the liver and the heart from the bats, and then we might use those tissues to extract DNA from to do genetic analyses. We use the specimens, the bones and other parts of the specimens for other uses as well. And so, this part of biology has been happening for a long time. And in fact, one of the reasons that Darwin avoided becoming a minister and went on to become the founding evolutionary biologist is that he took a voyage when he was a young man, largely to collect animals from South America. And he went with a Royal Geographical Survey ship and spent a lot of time in South America collecting all these extremely unusual, to his European eyes, specimens.
You describe or have described your work as transformational to this discipline. Explain how.
I'm not sure if I've used that word to describe it, but maybe other people have. So as a grad student, one of the good pieces of advice that I got from my mentor, Jack Sullivan, is that you shouldn't try to do work like other people have done it in the past, but you should try to understand why the work that they've done in the past might have flaws or shortcomings and then address those. And so at the time when I was in grad school and I was doing some of this work that we talked about earlier, this phylogeographic work, it was very common for biologists to use visual approaches for interpreting patterns of genetic diversity. And basically what they would do is they would make some sort of a figure or a diagram, and they would put that figure or diagram that was representing the genetic diversity within a given species onto a map. And then they would tell a story about how that pattern was formed. And so, for example, if you can imagine a species that lived in North America and most of the genetic diversity was in the Gulf Coast, they would tell you a story about how during the Pleistocene, species were restricted to this Gulf Coast habitat, and then they've gradually expanded since then, and that's why the genetic diversity is not very rich in the northern areas. This may be true, it may not be true, but it wasn't very scientific. There was little statistical approach to it. There was virtually no modeling. And so one of the things that I did as a grad student is I applied a model that was at the time new for how neutral genetic diversity partitions itself in a species. And this is a stochastic model, and the reason it's a stochastic model is because genetic drift is a stochastic process.
Stochastic meaning?
Meaning random, but following certain rules, I think, is maybe the easiest way to think about it. So basically, all individuals in a population and all populations within a species are related through this process of descent with modification from a common ancestor. And sometimes that modification happens due to processes like natural selection that are non-stochastic. But for most of the genetic variation, these are slight differences that are differences in the coding sites rather than the protein that those coding sites make. And because of that, those differences aren't really exposed to natural selection. So we call those sites neutral because you might have one, nucleotide base at a given gene at a certain site, and I might have a different one, but functionally they're the same because they make the same protein that does the same thing in the cell, right? And so selection that doesn't care about which of those you have, but you can use sites like that to understand the ancestry and the common ancestry of individuals and populations, et cetera. That can be done with a statistical modeling approach. And so I was one of the people that applied this modeling approach called the coalescent to this kind of data. And, I did that in part because it helped us to narrow down the range of possibilities that the history of a given species may have. So that's a bit of a tangent. But in essence, what that has helped our field do and people in my lab do is think about the historical data that we are using as the basis for inference in a way that's a little more rigorous and a little more scientific.
Can you still use that data, that historical
absolutely.
data you described?
Absolutely.
Even though it was not sort of collected or imagined scientifically?
So yes, and I think it's a difference in interpretation more so than anything else. One of the things that has changed a lot since I was a grad student is the sheer amount of those data have increased. We now live in a world where you can routinely sequence entire genomes from samples collected in the wild. I was kind of a hotshot as a grad student because I had multiple genes included. So I had, 2 or 3 genes in my dissertation. And my grad students look at this today and they laugh and they said, it would take you a long weekend to collect these data, right? You must have really worked hard for those 2 weeks you were in grad school with this data set you collected. And they just don't understand how much more laborious it was back in the day to do that. But now we have no choice but to use these modeling approaches because you really can't display a million variable sites on a map. It's just not physically possible to do.
Mm-hmm. You describe the biological sciences today as data rich. Is that because of genetic sequencing, because of our understanding of the genome? What's caused this, I guess?
I think 2 things have caused this, and they are both good examples of how investment by government funding agencies can really transform not only basic science, but applied sciences. And so it was part of the Human Genome Project that was, I think, established when Clinton was president. The U.S. made this investment to finally sequence every base of the human genome. And they started this project doing sequencing in the old way that had been revolutionary in the 1980s. And this method of sequencing was targeted such that you went out and you had to know a little bit about the portions of a gene sequence. And once you had that, you could make these little molecular scissors that would cut out that gene just for the animal of interest. And so in the case of humans, you would target a gene in humans, and it was one gene at a time. And you would gradually extend that by walking out from that one gene in the genome. Every 500 or 600 base pair fragment was a separate target, and you would have to design a separate set of molecular scissors to access that. And so it took thousands of technicians working like a decade or so to start to flesh out the Human Genome Project. As this was happening, a new kind of sequencing developed that was massively parallel and untargeted. And what this sequencing has grown into is a technology where my grad students can now go out and collect, hundreds of millions of sequencing reads with approximately the same amount of effort as it would have taken me to collect, a pair of those sequencing reads. And so, of course, this is why they look at my dissertation and laugh at it, essentially. But this investment by the U.S. government, and then later by other government agencies in different countries, led to this revolution in the sequencing technology, which has expanded the data that are being used by biologists. So that's one investment, and it's just technological and sequencing. But it forms the basis of everything from, the mRNA vaccines that helped us get through the pandemic to environmental, censoring techniques that people use for wild animal populations, where they go out and they collect scat and they sequence the scat to see which animals are present in the environment. So, wide-ranging applications for basic and applied sources that the people who did this project certainly didn't imagine. So the second big investment is an investment in the physical infrastructure that natural history collections have. And so we have millions of specimens in our little collection. If you go to places like the Smithsonian in Washington, D.C., or the American Museum of Natural History in New York City, they have orders of magnitude more. But all of these collections take a tremendous amount of personnel and care to properly curate and store those specimens. So in our museum, we have specimens that were collected, hundreds of years ago by people like Teddy Roosevelt, and they're in great shape. And the fact that they've been maintained for this long is a tribute to the professionals that we have working for them. Our curator, Tamaki Yuri, is incredible and has done great things with the collection at OSU. And there's thousands of people like this all over the world that are doing this hard work building and maintaining these collections. One of the things that the National Science Foundation has done is they have integrated all of the electronic databases that these different museum collections have. So if I have a student who wants to do research on a particular species, she is able to log into this database and find all of the specimens for that species in all of the different museums throughout the world, figure out which ones have tissue available, which ones have skeletal material available, in some cases, which ones have viruses that have been collected from the digestive systems of these species. And all of these data that are now data-based, and increasingly the data that are produced via scientific study from these specimens are accessible to anyone with a ability to log into the web. And so one of the things that my lab does is we do these large data science projects by downloading data from different natural history collections and then using those data in innovative ways.
When I hear the term molecular scissors, that puts me in mind of CRISPR.
Well, it's a related technique. CRISPR is a way of modifying existing DNA that is derived in part from this technique called polymerase chain reaction that served as the foundation for DNA sequencing in the late 20th century.
But you're not using CRISPR necessarily.
We're not. So the technical term is primer pairs, and what a primer pair is it's a stretch of about 20 nucleotide bases that fit into the reverse complementarity of the double-stranded DNA, and they can then excise that DNA, make a copy of it, and renature it so that it forms a new double-stranded copy.
But I did hear you say a moment ago that you use AI or machine learning in your lab, in your research?
We do. We actually have been early adopters of different kinds of AI approaches. So one example of that is actually something that we started during the pandemic. And I had in 2018, 2019, a pretty good run of luck with funding agencies, got some funding. I had recruited a bunch of really ambitious, excited grad students, and we were just raring to go. And then they shut everything down for a year. And, I was told, well, they're going to work from home. And if the way you work is hands-on physical specimens, that's challenging. So we ended up doing a lab project, and it was a project that was led by a graduate student, Danielle Parsons, who has since graduated. And what we were interested in learning is something about undescribed species diversity in class Mammalia. And I need to unpack that a little bit, probably.
Yeah, so, undescribed, that word is leaping out at me.
It is. So when we think of species, we often think of them in sort of the common language way, and we say, oh, there's a squirrel, or there's a deer, or something like that. But in fact, as we all know, there's multiple types of squirrels and multiple types of deer, et cetera. And the way that we know this is at some point, a scientist has investigated squirrels and figured out which individual squirrels were part of a single species group. And they have formally defined that species group using data from geography and genetics and morphology, et cetera. And, like anything in biology, this is not so much a hard and fast law as it is a best educated hypothesis type of guess. And so a species is described when someone has written a description of that species, given it a formal name and the Linnaean hierarchy, et cetera. And what we know is that since Linnaeus in the late 1700s started describing species formally, the number of species has grown, and it's grown because we've discovered new ones. It used to be that the way that this happened were, when people like Charles Darwin or Alfred Russel Wallace went out into the jungle of South America and they found things that no European scientists had seen before, and they described them as new species. And the way it's done now is perhaps not as exotic, but a lot of times it happens in museum collections where people look at material that has been collected and they realize that what we're calling the single species is actually two or more species. In those cases, those are undescribed species. And there's pretty good evidence that they should be described. But of course, it's laborious to sit down and look at every single museum sample from every kind of plant or animal and try to figure out where this description needs to happen and where this effort needs to be focused. So what my lab did is we downloaded essentially every piece of data that we could find on a public database from a mammal, so thousands of described mammal species. And we did some modeling with genetic data, with environmental data, and with some trait data, so things about the organism themselves, in order to make predictions about what types of species were likely to need species description. And this ended up being a very successful project. It was published in Proceedings of the National Academy, which is phenomenal for a first chapter of a graduate dissertation. It pointed to results that were not entirely surprising. And I found that actually fairly comforting because it suggested to me that taxonomists, the scientists who do the species description, generally have a pretty good idea about the work that they're doing. So small-bodied animals were more likely to have potentially undescribed species in them than large-bodied animals. And that's because, they're harder to examine, they're harder to look at, et cetera. Animals living in tropical or semi-tropical regions that had large geographic ranges. They're difficult to sample. They're also potentially variable across that range in ways that we haven't assessed yet. And so this was another indicator that we had. And it was a great pandemic project. It was a great way to use data science. The method that we used to make our predictive model was basically borrowed from Netflix. And it's a method called random forest analysis, and it's how Netflix decides what movies or shows to suggest for you to view. When you have an account and you've liked things and you've disliked other things, Netflix basically builds a customized predictive model that then spews out new recommendations. And so we did something analogous to that with this mammal data. And so that's one kind of AI that we use. We've also used other methods that are maybe a little more advanced called convolutional neural networks. And those are methods that basically find hidden and latent patterns in the data and then allow us to, do better jobs of classifying those data. And so I have a student who has used three-dimensional scans of bat skulls and AI models to extract data about differences between closely related bat species. And basically the technology that she's using is one where there's a CT scanner like you would have at a hospital, but it's a very small version. And we scan these skulls and make these huge digital files that are three-dimensional files of the very minute shape differences of the skulls. And then she has trained a convolutional neural network to extract thousands of different measurements in all different dimensions from these skulls. And that allows her to basically analyze morphological data that are approaching the scale of the genetic data.
How pervasive are AI tools or AI technology now in doing science? And I guess part of what I'm asking is that what it means to do science today? You do it with AI.
They're certainly not ubiquitous. I think those tools are more common than they once were. It is more common now for analytical methods to incorporate AI for particular kinds of analysis. But I think they're very much a continuation of the kinds of ways that biologists have always analyzed data. So if you talk to some people in computer science, they might make an argument that a principal components analysis is a type of AI. It's a rudimentary type of AI, but it's essentially reducing the variation of a data set using some predictable algorithmic rules. And so CNNs are more powerful, certainly, but they might not be fundamentally different. Random forests are more powerful in other ways and function differently, but, they're a continuation of some common themes that people have used.
This is a couple of years ago now. I was touring a chemistry lab, not here at Ohio State, elsewhere, and they were using automated processes. I don't know. It was a type of AI, I suppose. But they were talking about those AI tools almost as like lab members, in the same way that you would talk about the graduate students or the postdocs or something like that.
Well, I hope we never get to the point where my grad students and postdocs are AI tools, because I really like the interpersonal interactions that we have. We use those tools a lot for processing data, for collecting data, for, biology is heavily computational as an enterprise right now, particularly the kind that we do with genomic data, environmental data, and you just can't do it manually. So one of my grad students has a background in computer science, and, we tease Nico for not being a biologist, but at the same time, we're very grateful that Nico is in the lab because, he helps the biologists learn how to do some of this stuff more effectively.
How did you end up in biology? Did you know when you were a kid, when you were 4 years old, you were going to be a biologist, or is this something that came later?
I always liked biology. Like a lot of kids, I thought, dinosaurs were great. And, I like going in the woods and turning rocks over and stuff like this. I didn't expect that I could make a proper career of it or probably even think about it until at some point when I was an undergrad, I would guess. I majored in zoology and English Lit as an undergrad, which seemed reasonable to me. I've since learned it's a little unusual. But I kind of hate this narrative that an English degree is worthless, because I really use my English lit degree every bit as much, if not more, than my zoology degree in my day-to-day life as a professor of evolutionary biology.
How so?
Well, it's all, academia is mostly writing and reading. You learn how to do those things well if you have a degree in English.
Why bats? Why the particular interest in bats?
I think bats are fascinating animals, particularly because they're hard to understand. And I guess that makes them interesting to me and to many of the students that work with me. So they're animals that are very different in how they experience the world than we are as primates. And one of the things that's challenging about being a biologist is, the central piece, the central data point, the central way that we organize the world of biological diversity is by identifying different species. And what we hope to do is identify species that are equivalent to how the animals or plants identify another individual as being the same species or as being a different species, right? And we are primates. We're very good at visual interpretation and inspection. And it's natural that we try to identify species by visually, seeing similarities and differences. If you're an animal like a bat that might be really dominated by your hearing and your echolocation and probably your sense of smell, it's very likely that the cues they use to identify species are different than what we as primates use. And so this makes them challenging to study, but also I think very interesting. They're just, they're an amazingly diverse and successful group of animals that most people don't know very much about. And if they do know anything about them, they probably know the wrong things and factually incorrect things. So,
tell us what's next for your research.
So I'm really excited about work that a recent PhD graduate, Sydney Decker, has been doing in my lab. So Sydney's the one who's been doing work with these bat skulls. And one of the big challenges that we've had in evolutionary genetics is understanding how we can identify groups of individuals at the very earliest stages of diversification. And, in one way or another, a lot of evolutionary biology in the last 150 years has been about understanding how new species are formed. And the challenge with this is we often investigate units that are already so far along in this process that it's obvious that they're forming different species. And that gives us a certain conceptual challenge in discovering those factors that promote the earliest stages of diversification. So with genomic data now, we can increasingly identify groups of individuals that are at the very earliest stages of differentiation. But the question has always been, what do we do with these? What do we make of these? Because they're just slightly differentiated gene pools that in every other way are essentially the same, and it doesn't leave you a lot to question or to look into. So what Sydney's work with her yellow bats, which is a type of bat, is demonstrating is that if you collect high-resolution morphological data, you actually see that the patterns of differentiation in those morphological data are mirroring those that you see in the genetic data. And that gives us a much better way of looking at process. So in the case of the bats, it's probably has to do with morphologies related to feeding. And they're eating things that are slightly different, which are causing slightly different selective pressures in the size and shape of the musculature of their crania and their mandibles associated with that. And so this gives us new clues as to things we can look into and new ways of looking into those questions. And we need to do some diet analysis, obviously, and stuff like that. But we're starting to piece these things together. And the rest of the biological data is now starting to catch up to the genomic data in terms of the scale and how much of it we're likely to be able to collect.
Bryan Carstens, thank you.
Thank you.
Voices of Excellence is produced and recorded at The Ohio State University College of Arts and Sciences Marketing and Communications Studio. More information about the podcast and our guests can be found at go.osu.edu/voices. Voices of Excellence is produced by Doug Dangler. I'm Jen Farmer.